Greetings to everyone. In this video, we're going to discuss genetic engineering, which is an advanced field in biology with many applications in the fields of medicine, agriculture, and industry. So genetic engineering uses technology carried out in the lab to alter or modify the DNA of an organism, usually by inserting foreign genes from other species of organism into the host organism. So the end result is the production of genetically modified organisms with desired traits that can benefit mankind. Let's find out more about genetic engineering. So our learning standards are as follows. Firstly, we should be able to describe the terms genetic engineering and genetically modified organisms. Next, we are going to explain the application of genetic engineering in the production of genetically modified organisms, insulin, and genetically modified foods. So for this video, we're going to discuss up till number 1, right here, the production of genetically modified organisms. And production of insulin and genetically modified foods will be discussed in the next 2 videos. Here's the concept map for this chapter, chapter 13, genetic technology. So there are 2 aspects here we are going to discuss, genetic engineering and biotechnology. Let's look at the first part. So we're going to discuss the meaning of the terms like genetic engineering and so forth, and then applications. And we'll focus on the first one, genetically modified organisms. The other 2 will be discussed in another 2 videos. Firstly, we will discuss some important scientific terms like genetic engineering, recombinant DNA technology, GMO, transgenic organism, and so forth to help us understand this topic better. Let's find out the meaning of some terms. Genome. What is a genome? So a genome is a complete set of DNA from an organism that includes all the genes of that organism that is contained in the chromosomes, and that the genome contains all the information needed to build the organism and carry out its life processes. Okay, so here is a human being, and this, let's say this is a human cell. So inside the cell there's a nucleus, and the nucleus for humans there are 46 chromosomes, right? And on each chromosome there's a long strand of DNA. So the DNA consists of segments that form the genes, right? So a genome is the complete set of DNA for that organism. For example, for humans, that includes all the genes of the human, such as genes for eye color, genes for blood group, genes for height, and so forth. So the genome contains all the information needed to build the organism, the human being, and help it to carry out its life processes. Okay. Now, the fun fact is that the Human Genome Project was started in 1990, and it was carried on until 2003. It was an international research project that worked to determine the sequences of the 3 billion base pairs, as seen here, A with T, C with G in the human genome, and to identify all the genes that is contained in the DNA, right? So the findings that a person has about 20,000 to 25,000 genes in his DNA. 20,000 to 25,000 different types of genes in one cell, all right? And each person has 2 copies of that gene, one from each parent. For example, gene for blood group or gene for eye color. We have 1 copy of the gene. It's actually called the allele from our father, our own father, and 1 from our own mother. Okay. So genetic engineering is taking the world by storm with its many applications in different fields. What actually is genetic engineering? It is a gene manipulation technique where genes can be moved around, can be cut, can be joined together by the use of enzymes to modify an organism's genetic material artificially through man-made ways, not through nature, to produce new combination of genes. So to modify means to change the organism's genetic material in order to produce new combination of genes, and this produces a genetically modified organism called GMO, which has improved or beneficial traits usually. So genetic engineering doesn't really occur like what we see in this picture, where scientists use his hands to take out the genetic material, okay, or take out the bases or the nucleotides. It has to be done, the manipulation of genes has to be done at a microscopic level, and enzymes are used to cut and also to join certain genes or certain segments of DNA together. So two of the methods used in genetic engineering, which are very advanced, are recombinant DNA technology. Now, it's not called DNA recombinant technology, as printed in some books. It's recombinant DNA. Okay, the DNA comes after the word recombinant. So recombinant DNA technology involves the transfer of a DNA segment from one organism to the other, right? So that means we recombine the segments of DNA, recombine the genes again, as compared to the combination of genes in the original organism. Okay, so recombining the DNA again, and the transfer of DNA segments are carried out, okay, from one organism to another. It can be from the same species, from organisms of between organisms of the same species, but it can also be between organisms of different species. For example, bacterial DNA can be transferred to a plant. Okay. Now, another method used in genetic engineering is gene editing, such as the CRISPR technology, which is mentioned in the textbook too. So we have to know a bit of that about that. Gene editing allows scientists to add, delete, or modify, okay, to change specific genes within an organism's own genome, within the own genome itself, right? So in this case, only one organism is involved. So we are going to focus more on recombinant DNA technology and how it's carried out, with one example involving the production of insulin by bacteria. So that will be in the second video, okay? Production of insulin by bacteria. Next, what is recombinant DNA technology? So recombinant DNA technology is a set of techniques, modern techniques, advanced techniques used to manipulate, to move the DNA around. So manipulate and combine the DNA from different organisms or different species of organisms. So the DNA that is combined can come from two different organisms of the same species or of different species. By transferring the DNA or genes from one organism to another and combining the DNA of both organisms together, right? So the key thing is they transfer the DNA and then they combine the DNA together. Sometimes they combine the DNA outside the cell first and then they transfer the new DNA into the organism, okay? Whatever it is, when the two DNA from the two different organisms are combined together, you will produce the recombinant DNA, which is the new DNA with new genetic combinations, with new combinations of genes. So it's called recombinant DNA. The DNA has been recombined, okay, as compared to the original organism, which is with its own combination of DNA segments or genes, okay. So that's why it's called recombinant DNA, because the DNA strand that's produced consists of DNA from two different organisms. So it has new genetic combinations. And this will lead to the production of genetically modified organisms into which this recombinant DNA is placed, is inserted. Okay, so usually there's some beneficial trait that is produced by these organisms for the benefit of mankind. Now, 5 steps in recombinant DNA technology. First, isolate the DNA from the two different organisms. One is the donor, who is going to, or which is going to supply that gene or that segment of DNA to the other organism, which is the host organism. Okay, so isolate the DNA from donor, for example, the human insulin gene okay? And DNA from host organism in this case is a circular DNA called the plasmid, okay? The plasmid from bacteria. Now cut out the desired gene or DNA and cut the plasmid using restriction enzymes, not with a scissors, but with restriction enzymes that will carry out some chemical reactions and be able to cut out the desired gene from the donor, which is the human insulin gene from the whole long strand of DNA. And for the bacterial plasmid, you have to cut off a certain segment of this plasmid so that there's space for the new gene to be inserted into it, for the human insulin gene to be inserted into the plasmid. Okay. Next, join the DNA and the plasmid, the DNA from the donor, which is the human insulin gene in this case, and plasmid. Join them together, not with glue, but with DNA ligase enzyme. Okay. Then you see these red parts here are the parts where the two strands of DNA have joined together. Okay, so that's not very important. The important thing is the green part and the yellow part. They are DNA from two different organisms. And the whole structure here is called a recombinant DNA or recombinant plasmid in this case. Okay, with a human gene inside the bacterial plasmid. Next, insert the recombinant DNA into a host organism like a bacterial cell or a plant cell or animal cell. Okay, so this organism with the recombinant DNA is called a genetically modified organism. Next, clone the host organism. If it's bacteria, then we allow the bacteria to multiply, okay, by binary fission. That means the bacteria will divide and form more and more of the same type of cells, daughter cells that have this human insulin gene. Okay, so with this human insulin gene inside the bacterial cell, the cell will sort of automatically produce the protein. For example, in this case, it is the hormone insulin. Okay, so the host organism can be cloned, or it can just be allowed to undergo binary fission. Okay, a type of asexual reproduction in optimum conditions. Okay, then it will produce the protein like the insulin, which can be extracted for use. So this technology has many applications in medicine, agriculture, and technology. So we have already discussed what the meaning of recombinant DNA is. Now, for short answer, where you're asked to explain recombinant DNA, you can use the facts in note 1, number 1 here. But if they ask for more details, like the steps in recombinant technology, the general steps, then you can add in 2, okay, which is worth 5 marks. Okay, number 1 is worth 3 marks, number 2 is worth 5 marks. So what is recombinant DNA? Recombinant DNA is produced by recombinant DNA technology. It is a DNA strand that's produced artificially. Sometimes it's called rDNA. So this strand is produced artificially by man or by scientists in a laboratory using enzymes. And it combines the DNA from different organisms together, especially from two different species. For example, this is a recombinant DNA strand. It has the yellow, it has the DNA colored yellow here from species 1 and the DNA colored red from species 2. They have been combined together. This doesn't occur naturally in nature, right? Even through crossbreeding. So this is recombinant DNA that's produced in the lab, where scientists join two different DNA strands from two different species together using enzymes. Then this recombinant DNA strand is inserted into the host organism after it has been produced. So the host organism is the organism that will use this DNA strand, okay, perhaps to produce some new protein that it cannot originally do. Okay, so the host organism can be bacteria or plant or even an animal. And it will produce new combinations of genetic material in the host organism. Okay, this is a new combination of genetic material that uses DNA from species 1 and species 2. Then this host organism will have new traits that may be beneficial in some way, right? Now, recombinant DNA can be produced because DNA molecules of all organisms share the same chemical structure, where they have these bases like adenine, thymine, cytosine, and guanine. Okay, so this is very fascinating to know that all organisms share the same chemical structure in their DNA. All right, they have the same chemical structure, and they have the same types of bases like adenine, thymine, guanine, and cytosine. So they only differ in the nucleotide or base sequences in their DNA. All right, the base sequences are different. So that's why they can be joined together. DNA from different organisms can be joined together using enzymes. Now, the DNA used to produce recombinant DNA molecules can come from any species. For example, plant DNA can be joined to bacterial DNA. Human DNA can be joined to fungal DNA or bacterial DNA, as in the case of the production of insulin. The human insulin gene is joined to the plasmid from the bacteria. Okay, plasmid is the circular DNA from bacteria. Now, GMO or genetically modified organism is an organism such as an animal, plant, or microorganism whose genetic material or DNA has been modified or altered, changed somewhat in a laboratory through artificial means, using genetic engineering in order to change the organism's traits. So by by modifying the DNA of an organism, we can change its traits, right? Or give it a new trait that the original organism will not have. So GMO can be produced in many ways, and one way is by recombinant DNA technology, which we have discussed just now, which involves combining DNA molecules or genes from different organisms. Could be organisms of the same species or organisms of different species to create recombinant DNA, all right, which is a combination of two different DNA strands and with new gene combinations leading to the production of new traits in the genetically modified organism. So a GMO or genetically modified organism has new beneficial traits such as resistance to pests. For example, this type of corn that's called the BT corn can resist the pest, and the pest cannot eat it. If the pests were to eat it, like caterpillars were to try to eat the corn, they will die because this type of corn produce special toxins, all right, that can kill the pests. Okay. For bacteria that is genetically modified, there's one type of bacteria that is genetically modified so that it has the ability to produce human insulin. Okay, because it has the human insulin gene in it. So examples of organisms that have been genetically modified are rice, soybean, oil palm, pineapple, cow. Okay, there's a goat that can produce human clotting factor, and BT corn is corn that contains a gene for toxin production from bacteria. Let's find out more about BT corn. So let's look at this example of a genetically modified organism, BT corn. It is a GMO or genetically modified organism in which it has been inserted with a gene from bacteria, the bacteria Bacillus thuringiensis. Okay, so this corn here has received a gene from a bacteria, a type of bacteria, and this gene enables it to produce the toxins just like the bacteria does. Okay, so when the gene is passed on or transferred to the corn becomes a genetically modified organism, and with this gene for production of toxins from bacterium, it is able to produce the toxins because of the code from that gene. Okay, so it will produce the toxins which are poisonous to the pests. All right. Now, when it produces the toxins and the pest eats this corn, this type of corn, the toxin will kill the pests, such as caterpillars. So this makes the BT corn resistant to insect pests like caterpillars. And it will help to increase the yield of the corn because the corn will not be eaten by the pests, right? So the word BT stands for Bacillus thuringiensis, okay? Because the corn has received a gene from this Bacillus thuringiensis. Next, the term transgenic organism. It means an organism whose genome or DNA has been altered by the insertion of one or more foreign DNA from another species by using artificial methods, right? Like recombinant DNA technology. So transgenic organisms contain recombinant DNA, that is foreign DNA from another species, right? Inside the DNA, the foreign DNA from another species has been inserted. So that's why it's called transgenic, meaning trans here means across, okay? Transfer across. And then genic means gene. So the genes have been transferred across different species, from one species to another. All right? So it's called transgenic organism. Now, examples are BT corn, which has a gene from bacteria for the production of toxins. And then there's a transgenic goat that's been produced with a human gene for production of human blood clotting factor, right? This will be discussed afterwards. So are all GMOs transgenic organisms? No. Some GMOs are not transgenic organisms, okay? Because they are genetically modified, but they do not have a foreign gene from another species, right? So transgenic organism is just a type of GMO. GMO is a subset of the GMOs, right? So all transgenic organisms are definitely genetically modified, okay? But not all GMOs are transgenic. As in the case of an organism that is genetically modified by modifying its own genes, it didn't get any foreign genes from other species. That is not a transgenic organism, but it's genetically modified, okay? So transgenic organism is just one type of the many genetically modified organisms that are produced. I've already finished discussing the scientific terms related to genetic engineering. Let's go on to discuss some examples of GMOs, or genetically modified organisms. Here we see a glowfish, which has genes coding for fluorescent protein extracted from the jellyfish. So this glowfish has been inserted with the genes that enable it to produce a type of protein known as the fluorescent protein that helps it to glow in the dark. So this type of fish is sold commercially to the fish enthusiasts who like to keep this type of fish in their aquarium to rear them. Now, we are going to talk about two examples from the textbook, which are a cow, a genetically modified cow, and a genetically modified goat, which is a transgenic animal. So the first example of a GMO is the genetically modified goat, right? So it has the human gene which codes for a blood clotting factor. So inside the goat's DNA, in the original DNA strand, the human gene for blood clotting factor had been inserted inside, right, into the goat's DNA. This enables the goat to produce the human blood clotting factor that can help the blood to clot at a wound. So with this human gene, it's able to produce the blood clotting factor in its milk. Then this blood clotting factor is extracted and purified, and it can be used to treat the hemophilic patients who lack this blood clotting factor. So hemophilic patients suffer from hemophilia, which is a genetic disorder in which blood does not clot normally at a wound due to a lack of the blood clotting factor, specific blood clotting factor. So when the hemophilic patients suffer from a wound or a cut or a bruise, they can be given this blood clotting factor, right? And this product will prevent excessive blood loss for hemophilic patients. The second example of a genetically modified organism is a genetically modified cow that produces milk which does not contain the protein beta-lactoglobulin to avoid allergic reactions in children. So normal cow's milk contains beta-lactoglobulin, which is a protein that causes allergy, such as rashes and diarrhea in some children. So allergy means oversensitivity to a particular product, like for example cow's milk. Okay, in Chinese we say 敏感, and also in Malay we call it kepekaan, right? The person is too sensitive towards a product such as milk, and when the child who is sensitive to this product, oversensitive to this product, drinks the milk, right, he will experience rashes, diarrhea, and there may be other symptoms of the allergy. So this is sometimes due to the protein called beta-lactoglobulin in cow's milk, in normal cow's milk. By the way, this protein is not found in the mother's breast milk, right? So that's why children who drink cow's milk, they're drinking the cow's milk, they can experience allergy caused by the protein beta-lactoglobulin. Thus, the genetically modified cow's milk is different because it does not contain this beta-lactoglobulin to avoid allergic reactions in children, in certain children. Now, how did they produce this genetically modified cow? Scientists have added extra genetic material to the cow's DNA to stop the production of beta-lactoglobulin that is caused by a gene. That means it stops the gene from functioning and stops the gene from working to produce the beta-lactoglobulin. Now, how this process or technique is used is something different and new that we do not need to study, right? Just know that the genetically modified cow can produce milk that does not contain beta-lactoglobulin in order to avoid allergic reactions in children. Now, in the next video, we'll be discussing the production of insulin by using the genetic engineering technique. All right. So I will leave this for the next video. Please do keep a lookout for this video that's coming up very soon. All right. So thanks for viewing and goodbye for now.